I would now like to welcome Mohamed Nabil, who will be joining us from the Canadian University in Egypt. Mohamed, if you're ready for your presentation, if you could please turn on your camera and unmute your microphone and begin sharing your screen. Hi.
Okay. Hi. Good morning, good evening, good day for everyone. I'm glad to be here and thank you for your kind invitation for such a great committee. It's a great opportunity as I'm sharing my work, which is a part of my master's degree for fulfillment. So today I will gonna now share my screen.
We can see your screen, so please get started whenever you're ready. Thank you.
Okay, so the focus of my talk today will be about Adipose tissue-derived mesenchymal stem cells and their application in elevating or ameliorating the neuroinflammation and apoptosis events that mainly present in the aluminium-induced cognitive impairment model, which it is quite similar to the Alzheimer in aged people. So my talk will navigate, or I will navigate through the different sections, starting from the background and the hypothesis of my study, which led to the development of the aim of the work, and then talking about the methodology which I have applied to test this objective of my work, and the readout which I have selected carefully to test this objective, and showing what I have gained from results, and finally the end of this story or conclusion, what I have reached so far about this topic. So first I will discuss the background and the hypothesis of my study, and I will divide it into three main sections. First, I will talk about the aluminium neurotoxicity and its correlation with the development of Alzheimer's disease. Second, I will talk about Alzheimer's disease and give a brief info about information about Alzheimer's disease. And thirdly, I will discuss my approach of therapy, which is mainly regenerative approach. So aluminum neurotoxicity. As we know, aluminum, it is a common neurotoxic element which can be present in the daily life around us, as anyone can be exposed to it through inhalation in the environment or industry, or through food ingestion, or drinking water, or even taking drugs containing aluminum like antacids. So the problem here resides in the frequent exposure of aluminum can lead to intoxication inside the human body. And aluminum is considered neurotoxin due to the high ability of it to cross the blood-brain barrier, and this make a problem. As the aluminum can cross the blood-brain barrier, it can lead to the accumulation of amyloid beta and phosphorylated tau, which it is interestingly, they are the markers of, or they are the markers, the main markers, or the hallmarks of Alzheimer's disease. And after the accumulation of amyloid beta and phosphorylated tau, this will initiate a various or multiple degenerative cascades, including mainly neurotoxicity and inflammation, or apoptosis leading to neuronal death, or even impairment in the cholinergic transmission or oxidative stress. So what is Alzheimer? So Alzheimer is the most common form of dementia, as dementia can be caused by multiple factors. Most of it can caused by Alzheimer, accounting for about 60 to 70% according to the WHO annual report. So according to Alzheimer's Association, the early 10 signs of Alzheimer's it can involve memory loss that disrupted the daily life, like forgetting something have been learned recently, or changes in planning or solving problems, or difficulty in completing familiar tasks, or confusion with time or place, or trouble understanding the visual images and spatial relationships, or problems with words in speaking or writing, and this can take place in the advanced stage of the disease progression, or misplacing things and losing the ability to retrace steps, or decreased or poor judgment, or withdrawal from work or social network, or even changes in mood and personality, including depression. So what goes wrong inside the brain, or how it begins? So it begins when there is increase in a peptide or oligopeptide called amyloid beta. So the increase in the amyloid beta can be due to many reasons. The most common cause due to the decreased clearance as a result of the decrease of the secretase enzyme. So as the amyloid beta oligomers increase, this will lead to gradual deposition and diffusion as amyloid plaques. And the more of the amyloid plaques increases, this will lead to accumulation of amyloid beta aggregates. And this will lead to a sequential events, including stimulation of or increasing the tau hyperphosphorylation, inflammation, upregulating the inflammatory response, or even leading to neural and synaptic dysfunction. Overall of these sequential events will lead to overall cognitive dysfunction, leading to the signs and symptoms of Alzheimer's disease. Recently, or most importantly, neuroinflammation and apoptosis have gained a critical role or a major role in the progression of Alzheimer's disease. And recent studies have shown that therapies which are targeting neuroinflammation and apoptosis can be an effectuous approach or effective approach in the management of Alzheimer's disease and reducing the cognitive decline that present in Alzheimer's disease. So our approach was using the regenerative approach, which are mainly stem cell therapy. So stem cell therapy, especially mesenchymal stem cell therapy, have been a powerful tool and have again a wide array or a mark in the approach of treating multiple of various of disorders, especially or most importantly the neural disease, as shown here by the New York Stem Cell Research report, as it accounted for about 24.7% of the total research using stem cell as a treatment, mainly treating multiple of disorders, CNS disorders, including stroke, traumatic brain injury, learning difficulty, and Alzheimer's disease, or even Parkinson's disease. So mesenchymal stem cells can be derived either from bone marrow, adipose tissue, or umbilical cord. They can be either in vitro differentiated to the target cells, neuron-like cells, or in vitro expanded as it is undifferentiated, either that or that. They can be administered through many of routes. The most easiest way or convenient way, it is the intravascular route. After transplantation, they tend to home to the injured brain tissue and induce their effect either through neuron differentiation or either by paracrine action or reducing their paracrine action, which account mainly for the increased neuroprotection and increased neurogenesis and the immunomodulation. So the aim of our work was to investigate the potential ameliorative effect of adipose tissue-derived mesenchymal stem cells on the neuroinflammation and the apoptosis pathways, and see how the effect on these two pathways will affect or have implication on the overall effect of the cognitive impairment that present in Alzheimer's disease-induced animal model. So
Mohammed,
my method,
there's
okay,
5 minutes remaining. Just wanted
okay,
to
I
know
will,
that.
I will,
Thank
I
you.
will fast. Okay, okay. So my methodology tend to isolate stem cells from rat epididymal fat and characterize them according to the International Society of Stem Cell Therapy and expand them to 3 passages and inject them through intravenous route. After that, or after a period of time, uh, 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 I have done behavioral testing to see the effect on the behavioral action and the AD-induced rat, and furtherly doing, or at the end, doing histological and molecular analysis assay to test or the effect on the neuroinflammation and apoptosis and neurogenesis. So the readout which I've selected, or the biomarkers, mainly NGF. I have testing the expression level of NGF, which it is mainly for neurogenesis, and the anti-apoptotic PCL2 and the pro-apoptotic BAX, and finding the ratio BAX versus over PCL2 ratio, and the effect on the inflammation by determining the level of the pro-inflammatory interleukin 1 beta and interleukin 10. And most importantly, testing the level of the amyloid beta 42 through ELISA technique. So our result shows that the isolated cells from the rat epididymal adipose tissue tend to demonstrate all the mesenchymal characteristics as shown there: plastic fibroblast-like shape, and mostly the markers of CDs, they tend mostly to express CD90, and being negative for the other CDs, CD40, CD34, and CD45, and being to be able to differentiate into adipocyte and astrocytes and chondrocytes. Although the adipose mesenchymal stem cell transplantation have been significantly improving the cognitive impairment, this is present by the wastewater maze data test, as they tend to reduce the escape latency and reaching almost like the control group in the 3rd and 4th days. And the rats or the animals in the treated group tend to spend more time in the target quadrant compared to the untreated rats in the untreated group. As for the histopathological results of sections, the cerebral cortex of the ADMC's treated group tend to show almost intact neuron, as shown by the black arrows, with fewer degenerated neuron as shown by or pointed by the red arrow. While the AD-induced group cerebral cortex tend to show degenerated neurons with pycnotic nuclei, as shown by the red arrow. As for our markers, the ADMCs exhibits an anti-apoptotic effect. This is evident by the decreased ratio of PCL2 in the hippocampus and in the frontal cortex. As for the inflammation, the ADMC transplantation suppresses the expression of interleukin 1 beta and interleukin 6 in the hippocampus and in the frontal cortex. As for the expression of the NGF, the ADMCs modulate the, the expression of NGF in the hippocampus by upregulating it in the treated group as compared to the untreated group in the hippocampus, and even so in the frontal cortex as compared to the untreated group. All of these were consistent with the anti-amyloidogenic action of the ADMCs. As we see here, the amyloid beta level tend to be reduced in the treated group as compared to the untreated group, and slightly decreased in the treated group compared to the untreated group in the frontal cortex samples. So my conclusion, I have found that ADMCs could elevate the cognitive impairment caused by aluminium, at least partially through anti-inflammatory and anti-apoptotic, as well as neurogenic. And this tend to highlight the use of the or the application of adipose mesenchymal stem cells in the area of treating Alzheimer's disease in the next few years. And finally, I would like to thank Professor Hala Musallami and Professor Azza for the conceptualization and designing the experiment. And most importantly, I would like to thank Dr. Dina for helping me performing the experiments, analyzing the experiments, and mastering the technique of stem cell isolation. And I will not forget Dr. Mohamed Abdelrazak and Professor Said for helping us for analyzing the histopathological samples. So thank you for your time and waiting for your questions.
Thank you so much, Mohamed, for your informative presentation on Alzheimer's disease and for joining us from Egypt. I appreciate you being able to wrap up in the interest of time. And for the questions, I would just like to direct you to answer them in the Q&A chat function.
Okay.
So thank you very much for joining us today. And I would now like to welcome back Dr. Eleanor Fish to introduce our next keynote.